first I'd like to say thank you to the LRIG team and to AOMA for this opportunity. I really love these events, so I'm quite excited to present. So my name is Sasen Efrem. I'm a senior scientist at Ordeos, which is a startup at, it's in Soho. And this presentation is called High-Throughput Nanobody Design, Integrating Machine Learning with Rapid Self-Free Expression. Oh, perfect. I don't know if it's going to show up on the screen. I'll just use the mouse, but thank you.
You're welcome.
So just as a brief outline, what I'll be going over is Ordeos and the technology and strategy, our lab in loop, AI-derived binders against disease-specific targets, nanobodies and their advantages, as well as cell-free expression. I'll also be going over how we do rapid screening with high-throughput screening with highly correlated crude versus purified binding data, and some of our wet lab tools that we use at Ordeos. I'll also be going over some of our in vitro evaluations, so specifically cell binding assays, and how we can confirm binding in biologically relevant contexts. And finally, how we can engineer function with strategic multimerization, which is looking at bispecifics and multivalence to bridge targets or to increase avidity. So that's kind of briefly what I'll be talking about. So Ordeos reimagines biologics discovery as biologics design. So when I say lab in loop, this is kind of what I'm talking about. So you can see here that we start with design. We basically design binders in order to maximize in vitro success, and we do this using AI. And then from there, we actually make our binders, and we do this with cell-free expression. So we have a proprietary strategy to make these proteins. Cell-free expression is used by a lot of different companies or groups, but we kind of tailor it for our use. And it provides speed and flexibility. From there, we actually test the binders that are generated using high throughput methods to characterize and generate maximum data for optimization. And then we feed this back to our ML team. So they look at how well these binders performed. They look at different metrics, and they want to continuously drive improvement. So after that, once they have that data, they can redesign molecules based on that. And that's kind of the cycle that I'm talking about. So this is what I mean by lab in loop. Ordeos provides both nanobody and de novo protein solutions. So when I say nanobody, I'm talking about this here. So nanobodies, or VHHs, are these small proteins. They're about 15 kilodaltons, and they're the majority of what we make at Ordeos. So you can see on the left here what a conventional IgG is. It's about 150 kilodaltons, and it's kind of the typical antibody Y shape that you can imagine. Most mammals have antibodies like this. It's composed of 2 heavy chains, and then the light chain right here. So anything like rabbits, mice, humans, most mammals, like I said, have this conventional IgG. But what's interesting is that some camelid animals, like llamas, alpacas, and dromedaries, they have something called a heavy chain only antibody. So it's just the heavy chain right here. And actually the discovery of this heavy chain antibody in camelids I thought was quite interesting. So in 1989, there was a professor, his name was Raymond Hamers from the Free University of Brussels, and he was a biologist and a professor, and he was studying camels, so dromedary camels. He was studying sleeping sickness actually in these camels. And he was teaching a class, and in that class, he asked his students to do something very simple for most biologists. It was to use a protein G column and to collect the IgGs from provided serum samples and to separate the heavy chain and the light chain in a reducing gel. So they were provided with probably mouse serum or rabbit serum to do this, but some of the students wanted to be creative and went into the fridge and found some dromedary blood because the professor was doing this sleeping sickness study. And they decided, oh, I'll be creative and I'll do this exam with that blood instead. So they spun it down, they did the exam, and instead of getting the 50 kilodalton and 25 kilodalton expected bands, instead they got a very thick 45 kilodalton band. And they showed it to their professor and they said, this is wrong, there's no way. There's just no way that this can happen. Well, the professor couldn't live with this, like he had to test it again himself. So he tested it again, and lo and behold, that 45 kilodalton band was there. And the discovery was made that these animals only have these, or they're the only animals that have these heavy chain only antibodies. They have other antibodies, but this is the bulk of the antibodies that they have. And then from there, by 1996, another professor named Sergei Muldernis was able to isolate just this top part right here. This is the variable region, and this is the nanobody. And the reason why the nanobody is so exciting is because it's a single unit, it's small, and it's stable on its own, and it's able to bind to different targets just like an antibody. So immediately the potential for clinical use became very apparent. Sorry, I keep making sounds on this, but yeah, it was a very exciting time for the possibility of therapeutic use or even assay use. So yeah, this is the most of what we make. And then we also make de novo mini proteins, which are sort of custom tailored proteins for a given target. And both of these we make with machine learning. So what are the advantages of nanobodies, especially in therapeutics? So number one, genetic simplicity. So it is just a single cDNA that can be used to make the entire binding unit. And it's only about 400 base pairs. So for a regular antibody, you would need to co-express the light chain and the heavy chain. But for a nanobody, it's just very simple. It's just one clean DNA. It's very small, like I said, it's 15 kilodaltons, which can offer superior tissue penetration. So the potential to cross the blood-brain barrier and also to reach very small epitopes that maybe an antibody would be too big for. High physiochemical stability, so it can withstand extreme pHs, temperature, freeze thaws. It can rapidly refold, and it can be kept at room temperature. It's actually considered more stable than an antibody in most cases. It's low cost, and it's high yield with microbial production. So you can express them efficiently in E. coli and yeast and cell-free expression without the need for expensive mammalian culture or chaperones. You can also do modular engineering, which I'll talk about, which is basically, since you have these small units, you can connect them in interesting ways in order to do interesting things. For example, there's bispecific formats or albumin binding formats to improve half-life. So you can imagine that if you have a nanobody in the blood, it can be broken down pretty quickly. But if you have another nanobody that binds albumin and you link them together, that actually improves the half-life because it's kind of being carried by the albumin. And another example is here, caplacizumab. And this is the first FDA-approved nanobody. And this is just a single unit, and it's actually used as a bivalent homodimeric nanobody. So it's just the same unit connected to itself, and this increases avidity. Like I said, there is a regulatory proof of concept. So FDA approved caplacizumab shows nanobodies can meet safety, efficacy, and manufacturing standards. So this is sort of a visualization of what in silico a nanobody might look like. So you can see that this is actually the unit right here, and the blue is the scaffold. So the scaffold is composed of these 4 frameworks, and we actually have a fixed framework that we use for the majority of our nanobodies. So these are unchanged, we don't mutate them. And then from there, we have the 3 CDRs, with the yellow one, CDR3, being the most important one involved in binding. So it's typically the longest CDR, it's the longest section that we change, and it's the section that has the most variability and the most mutations. So these are the things that we play with ML in order to pretty much bind any target, just how an antibody can pretty much bind any target. And then at the end here, we have our tags. So we have a triple HA tag. We use this for SPR, and then C tag, which is what we use for purification, but recently we've changed the C tag to a strep tag. So in terms of cell-free expression, what is that exactly? So it's something that's very simple. You have a tube, you have E. coli, or you can even use mammalian cells. You grind them up, and because the cells have all of the components that are required for making proteins, the lysate of these cells can be combined with an energy source, cofactors, amino acids, NTPs, DNA, and a few other ingredients, and you can actually just make the protein in a tube, which is awesome. It's from -80 degrees to functional protein in less than 2 hours. So when I say that, you can pull the DNA from your freezer, pull the lysate from your freezer, mix them together, and in 2 hours you will have the protein made, which is very different from culturing mammalian cells or culturing E. coli cells, which can take days. So 60 to 90 minute expression run, complete freezer to protein cycle in under 2 hours, cost efficient and in-house controlled. So this reduces per sample costs and gives greater flexibility to optimize reaction conditions. So we buy all of these in individual components separately, which makes it much more cost efficient than buying a kit for this. You can also engineer your E. coli lysate for your specific needs. So we have a strain of E. coli that we have engineered so that it has a certain component removed, which makes our purification easier, for example, but you can also engineer them for high soluble yield, which we also do. Minimal reagent needed, so we do 20 microliter reactions for screening, and this makes just nanograms of proteins, which is more than enough for SPR screens, which I'll show. We also can use there's flexible DNA templates that can be used. So it accepts both linear and plasmid DNA. You can also handle difficult targets, for example, toxic proteins that might otherwise kill cells. You can imagine that obviously in a cell-free environment, that's not gonna matter. But if you're trying to produce a toxic protein in a cell environment, it would just kill your cells and end the reaction. So yeah, you can make toxic or aggregation-prone proteins without the constraints of live cell growth. And finally, it's easily scalable and automatable. So you can do 2 to 50 microliter reactions in 96 or 384 well plates, and it is compatible with liquid handling robots because, again, it's just mixing reagents. So this is just sort of a proof of concept of caplacizumab in our lab. So you can see here, this is our gel. And on the left, this is caplacizumab produced mammalian ladder, and then this is caplacizumab produced with cell-free expression that has been purified. So you can see they're relatively close. mammalian is just glycosylated. And then from here, you can see if we load caplacizumab, wash and apply the target for caplacizumab, which is von Willebrand factor, it shows clear binding. So this is just showing that these binders are properly folded, disulfide formed, and functional. So I'm a scientist, so the ML side is not really my wheelhouse, but I can briefly go over the strategy for the ML side of things. So we basically start, so this is for completely de novo proteins. We do sometimes start with a lead and we optimize off of that, but when it comes to completely de novo, we start with broad design, so structure native de novo generation, and this is basically just diffusion. So we're using diffusion to create out of nothing something that looks like a real biological nanobody. And this is based on just tons of data, tons of basically databases of existing antibodies. You just want to create something that looks like an antibody, that looks like it'll fold correctly. And you can see here in silico what that looks like when you're trying to make it an appropriate antibody that you want to land on. And then from there, you optimize, so you can do alanine scans, you can do large language mutations in painting. This is basically diversifying your initial diffusion lead. And then you can do a series of preps, so you can do complex docking, MD simulations. We run a lot of binding models to see how well this large set of design binders would likely bind to our target. And we also run some models to see how well they would meet our QC metrics. So we want to make sure our binders are stable, well folded, and not likely to aggregate. And then once we have a subset of, let's say, 10,000 or whatever number, 100,000, we'll take the best 200 and I'll test that in the lab. So then that will go to the lab over here. And this is when we actually test them and feed that back into the ML team. So you can see we design the proteins. We land, so in terms of SPR, and SPR is just looking at how well it binds to its target. We might land, let's say, 2. Let's say we have 2 that somewhat bind to its target, but then we have, a handful that meet QC metrics and we really like the way that they fold. Then we continue to train, and then we expand off of the ones that were positive. And then the next round, we hope to see, a lot of binders that are even better than the previous one, and a lot that are even better in terms of QC. So that is the kind of land and expand strategy that we use. In terms of our workflow, you can see how that looks. So once we have our sequences selected, so this normally takes hours to days to actually design the sequences, we send them to IDT to make the DNA. So we normally do 2 plates at a time. We get 188 samples. We save some wells for controls, so normally 2 per plate. And actually our biggest bottleneck is just waiting for the DNA to come back from IDT. Then we run it, we use the Cartera for high throughput screening. This is kind of my forte, that's the thing that I do. So we run this to see if it binds to its intended target specifically. I guess the step before was to actually make it. So once we have the linear DNA, we do cell-free expression, which takes 2 hours, and then we run it on Cartera. So this is basically like the step here when we actually receive the DNA, cell-free expression, and Cartera, that happens all in one day. So by the morning, we start cell-free expression. By around 30, we might start the Cartera run, and then by the late evening, we'll have some understanding of whether these binders bind to its target. And then from there, the ones that we like, we can re-express in cell-free expression at a larger scale. we can purify them, and we can do some QC. So we can do things like DSF, SEC, SDS page. We can rerun them on Cartera to see if they still bind once they're purified, how it compares to the crude version. And then we can, for the ones that we really like, we can do function. So we can look at cell binding in flow cytometry. But most of the time, once we see the binding here, we go back to the ML and continue to iterate until we're very happy with our binders. This is just to show that our crude lysate screening accurately predicts purified protein binding. So we have a very strong agreement between our crude proteins and the purified proteins. So again, the crude protein is when you have your lysate and you express your protein and it's just sitting in the lysate versus actually purifying that out. So when you compare binding between the two, it's very, very close. So the lysate itself doesn't have much effect on the SPR that we do. So this high correlation means that we can rank candidates directly from crude reactions and then purify only the top hits for confirmation. And this validates crude lysate SPR as reliable, high throughput, and a great strategy for our ML-driven loop. And as you can probably imagine, this saves us a lot of time and materials. So this cuts days and consumables out of each design cycle because you don't have to purify every single binder that you're testing. You know, if you have 2 plates and you have 0 binders, there's most of the time no need to purify that unless you're interested in things like how well they produce and how stable they are. And then quickly, this is just to show some of our cell binding assays. So you can see that this is using flow cytometry. These are 2 binders that we really liked and we looked at in terms of cell binding. You can see that among binders that pass QC thresholds, candidates advance to cell binding assays to determine whether binding is also seen in vitro. So it's basically just to prove that, okay, it binds to its recombinant target. Let's see if it actually binds to the same target in cells. And we're able to see that, at least in this case. And then finally, strategic multimerization. You can see that you can do different things and have different modalities with these binders. So you can either have 2 different binders, so this is a bispecific. You can have an FC domain, and that might allow for 4 different binders, and to really increase the avidity that way. So in cases where bridging of multiple proteins is required for function, we can engineer multivalent molecules. So some reactions in cells in biology actually require like 2 proteins being pulled together, like for example on a cell membrane. And so you can engineer these nanobodies to be linked in a way that it helps bridge them together, holds them together. That's one thing you can do. And nanobodies are also selected with kinetics matched to cell surface target density. So if there's one protein that's super ubiquitous on a cell, you probably don't need that high of a affinity to bind to that. But then if there's another protein that's way more sparse, you want to really make sure you're holding on to that. So that one would require a much higher or much better KD. And so it's not always the case that you want to necessarily optimize for the best and lowest possible KD. It's just the most appropriate for the given context and the given biological function. So these linker configurations are often guided by in silico modeling. And this is just some of our alpha LISA data that we use in order to show function with our cells. So in summary, we do ML guided binder design. So this is rapid generation of high affinity candidates against disease targets. We do high throughput cell-free expression to produce and screen lysates at the microliter scale. We do high throughput SPR to identify hits that have a high crude to purified correlation. We use QC to evaluate stability, producibility, and aggregatability of these binders. And we do in vitro binding validations to determine cell binding activity and to prioritize binders that have high therapeutic potential. We also can do functional engineering to optimize via multimerization or fusion strategies to enhance avidity or mechanism-specific activity. And that's it. Thank you.